Super-resolution structured illumination microscopy, 3D SIM, is an innovative technique for the visualization of fluorescent reporter proteins, of germination receptor clusters, and spore membranes. Using these procedures, unsurpassed resolution of germinosome localization and spore inner membrane lipid domains can be achieved. The technique will be demonstrated by PhD student Juan Wen and Master of Science student Raymond Pasman of our laboratory.
Before beginning the procedure, clean high-precision cover slips with 1 molar hydrochloric acid for 30 minutes in a gently shaking water bath, followed by 2 5-minute washes in ultra-pure Type 1 water. After the second wash, place the cover slips in 100% ethanol for about 3 minutes, before drying the cover slips and verifying their clarity. Then clean glass microscope slides with 70% ethanol for 1 minute before allowing the slides to dry and verifying their clarity. For fluorescent microsphere and spore imaging, first pre-warm 2 slides on a 70°C heating block for a few seconds before adding a 70°C 65 microliter droplet of sterilized 2% agarose onto one of the slides. Place the other slide on top to spread the agarose between the slides. After 5 minutes, remove one of the slides and cut the dried agarose into a 1 by 1 centimeter section. Add 1 × 10⁸ fluorescent microspheres or spores in 0.4 microliters of sterile ultra-pure Type 1 water to the agarose, and place a high-precision cover slip onto the patch, using the cover slip to slide the patch off of the slide onto the cover slip surface. Then, fix a 65 microliter 1.5 by 1.6 centimeter gene frame onto a dried slide, and place the cover slip onto the frame, closing all of the corners of the frame. For imaging of the samples, place the slide onto a structured illumination microscope equipped with a 100x oil objective, and focus on the 100 nanometer fluorescent microspheres. Adjust the correction ring on the 100x objective until a symmetric point spread function is obtained to minimize blurring of the images, and select a field of view with approximately 10 round fluorescent microspheres. Apply a grading focus adjustment for the designated excitation wavelengths, in this case 561 and 488 nanometers, as a guide for the image analysis software before focusing on the spores with the transmission light. Capture a transmission light image in the 16x average mode with 20 millisecond exposures for each image. Then capture 3D structured illumination microscope raw fluorescent images of the spores with the 3D SIM illumination mode. For slice reconstruction, click Param on the Structured Illumination Pad tab sheet to open the NSIM Slice Reconstruction window. Follow the suggested instructions and click on the appropriate controls to set the illumination modulation contrast to auto, the high-resolution noise suppression to 1, and the out-of-the-focus blur suppression to 0.05 as starting points. Then click Reconstruct Slice to reconstruct the image and evaluate the quality of the reconstructed images by the fast Fourier transformed images and reconstruction score that are displayed after the reconstruction. Adjust the high-resolution noise from 0.1 to 5 and the out-of-the-focus blur suppression from 0.01 to 0.5 until the best parameter settings are obtained. Then click Apply to apply the changes. Click Close to close the window. Open an FM464 stained PS4150 spores raw image. Then click Reconstruct Slice to execute the slice reconstruction and save the reconstructed image. To convert the 3D SIM raw images of the KGB80 germinosome into pseudo-widefield images, left-click on the ImageJ SimCheck plugin and select Raw Data SI and Pseudo-Widefield. Randomly select about 25 spores in each inverted transmission image for later germinosome analysis in the fluorescent pseudo-widefield images, until approximately 350 spores have been selected. Then use ImageJ to assess the maximum intensity for each fluorescent signal expressed by each group of spores, and the integrated intensity of each 3D spore image. To analyze the pseudo-widefield images of the KGB80 germinosome, use the mean integrated intensity value of 7 stacks as the integrated signal intensity of the KGB80 spore. Then determine the background intensities by imaging the PS4150 background strain using identical settings. And regard the fluorescent spots in individual KGB80 spores as germinosome foci when they are clearly distinguishable from the background. In this representative experiment, 2 foci of the GER-D GFP fluorescing spores appeared in different stacks, with 3 total GER-D GFP foci observed in the compositive Z3 stack. Here, a spore with only 1 GER-D GFP focal point in the spore was observed. In total, around 40 to 50% of the spores had 2 or 1 GER-D GFP and GER-KBM cherry cluster respectively. While the integrated intensity of the GER-D GFP scaffold protein was different between different populations, the integrated intensity of GER-KBM cherry was about the same in different populations. When the spore had multiple foci, the maximum fluorescence intensity of GER-D GFP and GER-KBM cherry foci tended to decrease, and the maximum fluorescence of all of the bright spots, regarded as the germinosome foci, was higher than the maximum autofluorescence of the PS4150 spores. Notably, brighter FM464 spots similar to germinosome foci appear in both intact and decoded PS4150 spores, suggesting that these brighter FM464 spots might be involved in the clustering of germinosome proteins in the inner membrane.
Adding spores to the agarose and placing agarose into the frame requires a continuously and careful fluid movement for these minute materials.
The conversion of 3D SIM raw images of the KGB80 germinosome into pseudo-widefield images and their interpretation requires expert knowledge on spore biology.
Our structured illumination microscopy procedure can be applied to the imaging of low-abundant proteins or dim fluorescence reporters in different bacteria or other biomaterials.
The organization of the germinosome can now be studied using double labeling procedures to assess co-localization of bacillus germination proteins and specific membrane domains.